So far in chapter 36, we've been talking about flow of water across a membrane, across aquaporins, following solutes, for instance. And that's fine and dandy, but that doesn't go very fast. So if you want efficient transport of fluids across a long distance, you need bulk flow. So this is essentially flow driven by pressure through tubes, and plants do this. Tracheids, vessel elements of the xylem, and then the sieve tube elements of a phloem allow for bulk flow through, in the xylem's case, cells that aren't alive anymore, that have sizable tubes that use the cohesion and the adhesion of water and capillary action to move water up a long distance. Using the xylem, plants can move a huge amount of water from the roots in the ground to the shoots that might be a few hundred feet in the air if we're talking about, for instance, a redwood tree. At first, water is absorbed by the root tips, specifically where the root hairs are, because that's where you have a whole lot of surface area. And that allows water to move into the root cells, or at least through the apoplastic route, into the extracellular space where the cell walls are. And then once we enter the roots, there's lots and lots of surface area again, full of the cortical cell membranes. It allows you to uptake water and particular minerals, and then you have to move that into the water cylinder in the middle of the roots to allow it to move up the xylem. And the concentration of solutes and minerals is greater in the roots than in the soil, partly because of active transport. Plants will expend energy to move minerals and solutes into the roots, and then water follows that. As water moves in, it eventually runs into the endodermis, which is the innermost layer of cells in the root's cortex, and it's around the vascular cylinder. And it's a checkpoint to make sure that you're not bringing things into the vascular cylinder that you don't want to bring in. And it's able to be a checkpoint because there's a waxy strip called the Casparian strip that goes around the endodermal wall, and it blocks anything that typically could flow with water straight into the xylem from moving in without actively crossing a plant cell membrane. So if we zoom in on this little root hair here, water and minerals can come in either through the apoplastic route or the symplastic route. And in the symplastic route, they've already gone through a cell membrane, and so there's been some measure of control over what comes through, and they can kind of go just directly through this cell membrane here. But if they go through the apoplastic route, then eventually they're going to bump into this waxy strip here, and they can't just go across the waxy strip. They have to zoom over here, go into a cell across the membrane before they can get into the xylem. All this means that for anything to get into your xylem in the vascular cylinder, it has to first cross a cell membrane, and that gives you some control, you being the plant, of course, over what gets into the xylem. So now the water is inside the xylem, and we call this water and the dissolved minerals associated with it xylem sap, and they are bulk transported from the roots all the way up to the leaves. And this involves a process called transpiration, which includes the evaporation of water from the plant's surface as more water moves up behind it. But how does this work? Is the sap pushed up from the roots, or is it pulled up from the leaves? And the answer is a little bit of both. So because we're pumping ions and mineral ions into the xylem, the vascular cylinder, lowering the water potential, which means water then flows from the outside of the roots into that vascular cylinder, that creates some positive pressure that pushes water up just a little bit. And so a lot of plants, especially shorter plants, will end up in the middle of the night when there hasn't been a sun to evaporate the water. You'll see on the tips little droplets of water, and this is called guttation, and it's a result of root pressure, which is this pushing up of water from the roots. And this is different from dew. Dew has to do with the humidity and differences in temperature between the air and the inside of the plant cells. And like when you put your cold Coke can out on a warm, humid day, water will become liquid from the gas in the atmosphere around the outside of your can. This is different. As you can see, the water's only on the edges of the leaves, at the ends of where the veins are, the vascular cylinders. And this is because water has been pushed up from the roots, and it drips out here just a little bit. Again, this is called guttation, and it's from root pressure. That said, while this does happen, it's a relatively weak force in terms of bulk flow of fluid through the xylem. The bigger force has to do with transpiration. And it's explained by this cohesion tension hypothesis, which is that water continuously pools on the water molecules below it because of the polarity of water. Water has polar covalent bonds. So the oxygens are a little bit negative because electrons from the covalent bonds spend a little bit more time by the oxygen, and they're a little bit positive by the hydrogen. And so the oxygen of one water molecule is attracted to the hydrogen of the next water molecule. And so you can get these chains of water molecules that are attracted to one another. This is called cohesion. They're also attracted to the sides of the tubes, the xylem tubes that they're in, and that's called adhesion. And that allows some capillary action, allows the fluid to flow up a little bit. But mostly you have water that's evaporating from the leaves, and as it moves up, it pulls on the water molecule below it, which pulls on the water molecule below that, and below that, and below that, and you end up with this big chain that pulls water up from the bottom. And this is really the bulk of the pressure, negative pressure pulling water up rather than root pressure, which pushes up from below. This is called transpirational pull. So in the leaf, water will evaporate, creates water vapor, and that creates a potential, right? It creates sort of a vacuum, and it pulls up the water below, creating a negative pressure potential, pulls the water up into the mesophyll cells. Here you can see the water beginning to evaporate, which creates more surface tension up here because of the cohesion of water, and that pulls up water below it, right? Which pulls water up from below it, essentially all the way through the xylem from down to the roots. And this process, which again results from the cohesion and adhesion of water molecules because of their polarity and their hydrogen bonding, results in a lot of negative pressure up here from the evaporation of water that pulls water all the way up from the roots, sometimes a few inches or a few feet, or in the case of giant redwood trees, again, we're talking about often a few hundred feet. This is a BioFlix video that you can find on your Mastering A&P, and I highly recommend watching it if you're having trouble with this idea of bulk water transport. In order for this all to happen, you have to have a continuous column of water from the bottom to the top, and you need to have strong vessel walls that can deal with that negative pressure, which plants do have because of the lignin in the secondary cell walls of the tracheids and the vessel elements. But if there is a lot of drought stress or freezing, it can cause those columns to break, and that is not good, and it causes a little noise. It's called cavitation, and I've been told that if you have the right equipment, something like a stethoscope, right, you can actually listen to on a very dry or cold day, the breaking of little water columns in a tree. To review, this is all maintained by the transpiration cohesion tension mechanism, meaning at the top in the leaves, you have evaporation. So this energy is coming from the sun, causing evaporation of water molecules, and that creates a water potential difference, essentially a vacuum, and it pulls water up, and water, because of its cohesion, can pull up other water molecules all the way from the bottom of the roots. And this is different from diffusion. It doesn't have anything to do with solute potential or gradients of concentration. It has to do with pressure differences. And it goes much faster, much larger amounts, and it moves not just water or solutes, but the whole solution up those hollow dead cells. Plants can regulate how quickly this happens based on little mouths that we talked about called stomata, or stoma singular, in the leaves. And these stomata have guard cells, which can, again, open or close around the hole. And if they're open, they allow more evaporation to occur. More transpiration means more water coming up. And if they close, it means less transpiration occurs. Here you can see turgid guard cells, which means an open stoma. And here you can see a closed stoma with less turgid guard cells. Of the water that comes up the xylem, some of it is used for photosynthesis, some of it is used for filling up new cells in a plant, but the vast majority, 95%, is lost through the stomata through evaporation. So again, it's very, very important to be able to control this using the guard cells. Guard cells that have filled up with water and become turgid bow outward, and that space between them opens up. And when those cells are flaccid, they become less bowed and the pore closes, the stoma closes. And this level of turgidity can be controlled by pumping ions, specifically potassium ions. If you pump potassium ions into this cell, water follows through osmosis and they become turgid and it opens. If you allow the potassium to leave, the water follows and they become flaccid and the stoma closes. Environmental conditions can affect whether a plant will open or close these stomata. Generally, they're open during the day because that's when photosynthesis is going on and you need to allow CO2 in and allow oxygen out so that you don't undergo photorespiration, which inhibits photosynthesis. You may remember from Bio 131. What causes them to open during the day? Well, light can cause them to open. The lack of CO2 can cause them to open. And a lot of cells have an internal clock that causes them to close or open, specifically based on a circadian rhythm, a 24-hour cycle. Also, drought or extremely high temperatures or wind, which all can exacerbate evaporation of water, can cause stomata to close even during the daytime. Because 95% of the water leaves, plants lose a lot of water through transpiration, and if it's not replaced, plants will lose water, they'll wilt, and they won't be able to function as effectively. And eventually they'll die. And so it's very important for plants in dry areas to be able to conserve water. We call plants that are adapted to arid, dry climates xerophytes. Xerophytes often have adaptations like really thick cuticles to prevent water loss. Or they may have very, very tiny leaves, again, waxy, with wax produced by the cuticle. Sometimes the leaves are even adapted to be really thin like this, or like you might find on a cactus, so that they don't have very much surface area to lose water. A lot of desert plants have very short life cycles that can be completed entirely during the rainy season. So you essentially don't see them during the summer or winter, but only for a brief time during the spring. Many of them can even leave their seeds in a seed bank until you get a particularly rainy spring. And you actually, during a rainy spring, you get beautiful blooms of flowers in the desert. Others have modifications in the way that they photosynthesize. We talked last semester about CAM plants, CAM photosynthesis or C4 photosynthesis, which are ways that you can keep your stomata closed more than typical and still avoid photorespiration. Phloem sap is very different from xylem sap. It is really, really high in sucrose. So think about like your maple sap that you make syrup out of. And it tends to travel from a sugar source where you make the sugar or where you stored it to a sugar sink where you're going to use that sugar or possibly store it. Storage organs like roots in a carrot or a sweet potato, for instance, can be sugar sinks in the summer when you're filling them up with sugars, and sugar sources in the winter and then spring when you want to grow a new plant. And you can move sugar through the symplastic or apoplastic pathways. This is an active process. We actively move sucrose across a membrane by pumping hydrogen ions out, and then, as we talked about earlier, they flow back in and pull in the sucrose with them. And once you've pumped sucrose across a membrane, water follows through osmosis. So using hydrogen ion cotransporters, we pump sucrose ions across a membrane, water follows, and so we end up pushing through positive pressure in phloem sap instead of pulling through negative pressure like we tend to with xylem sap and transpiration. The source will make sugar, we'll pump it into the sieve tube element, water will follow, positive pressure pushes it down or up, but it pushes it toward the sink, and in the sink we pump the sucrose into the sink and water follows, but then water is recycled back into the xylem. Leaves tend to be sources, fruits or flowers or seeds tend to be sinks, and storage organs can be, again, sinks in the summer or sources over the winter and in the following spring. So we don't always move up or down, we move from source to sink in phloem.